Welcome. I am Bill Powell, the Director of the American Chestnut Research and Restoration Project here at SUNY ESF. Today we're having a program on how to pollinate the American chestnut tree. This is a program so that you can actually help with the restoration. When we made a blight-tolerant tree, we basically made a clone to start with, but we need to outcross that clone to as many trees as possible, and this is something you can do. This program will train you on how to take our transgenic pollen and cross it with wild-type American and actually produce offspring that will be more diverse and ready for our restoration. So I hope you will enjoy this program, you will learn a lot, and you can help us out.
Welcome. I'd like to give you some background on our American Chestnut Research and Restoration Project, and specifically about what we're doing with pollinations and why. So there are a number of reasons why people might want to pollinate chestnut trees. We are specifically interested in propagating our transgenic American chestnut that was developed here at ESF. It has a gene for oxalate oxidase, and that allows the tree to tolerate chestnut blight. It is unique in that it doesn't kill or repel the blight fungus, so we talk about tolerating the fungus instead of resisting it. I'm not going to go into a lot of detail about the transgenic tree, but we have done a variety of safety tests, or what we'd call risk assessments. Those are described elsewhere. But the main point of our pollination efforts is that genetic diversity is absolutely crucial for longer-term restoration efforts. And with healthy natural populations of chestnut trees, there are a variety of traits and mutations and adaptations that make these populations adapted to their natural environment. And some of these differences are regional, that might help them uniquely adapt to where they are growing. But also it's important to think about resilience and possible future threats. There might be other diseases that we don't even know about yet, or environmental threats. And so having a diverse population is really important to be able to be resilient to some of these future threats. With our transgenic tree specifically, we are interested in increasing genetic diversity, and the main way that we are doing that is through outcrossing or pollination. And we talk about outcrossing because we're incorporating genetic diversity from other species outside of those that we have worked with in the lab. And so we are crossing out to some of these wild surviving American chestnut trees that still persist in the wild. We've been working closely with the American Chestnut Foundation, and some of their genetic models are showing that on the order of hundreds of unique sources of American chestnut trees over the course of about 5 generations should result in a diverse and resilient and adaptable population that would be appropriate for restoration. I'd also like to mention inheritance of the OXO transgene, and it is important to remember that about half of the offspring from a cross with the Darling 58 chestnut tree will inherit the transgene. The other half don't. So they're non-transgenic trees. This is important for potential future efforts that might be even better than the current efforts we have now, or for kind of continually having non-transgenic trees in the population. It's also important to remember in inheritance that we need to identify which trees, which offspring trees have the gene and which do not. So some of those identification methods will be described elsewhere also. There are 2 main goals for diversity and for outcrossing efforts. The first is restoration, like I've mentioned, and this is going to take a while. Like the 5 generations that I mentioned might take up to 20 years. But in the shorter term, we're thinking about things like backyard trees or horticultural trees. These won't be quite as diverse as the restoration population, but they still have some diversity, more than the initial tree we developed in the lab, and they should be available much sooner, potentially as soon as we finish working with the regulatory process. Speaking of the regulatory process, everything we do right now with transgenic pollination or with transgenic trees has to be done under permits. So we are not able to share transgenic pollen at this point. We can't send it or use it outside of our permitted plots. But we are working with all 3 regulatory agencies in the US, and we're hoping, if things go smoothly, that within maybe a few years we should be able to start distributing some of this transgenic pollen. So now is a fantastic time to practice pollinating and to have a lot of people be good at pollinations. Now, catkins, or flowers on American chestnut trees, are up high, and so of course we need to reach them to do pollinations. But it's important to remember that lifts and ladders, while essential for pollinations in many cases, can be dangerous. So please be careful. The most important thing, don't work alone. Have someone else nearby. And in some cases, especially mechanical lifts, might require training before you can use or rent them. But there might be some creative alternatives. People are starting to think about using drones for pollination, or if you're able to contact a trained tree climber. I've heard about people building scaffolds around uniquely valuable trees. So be creative and be careful. I'd like to wrap up by emphasizing that restoration will absolutely depend on you, on the public, on chestnut enthusiasts, on the American Chestnut Foundation has been an incredibly valuable partner, and they will be probably heading up our restoration efforts moving forward. And thank you very much for your support. This restoration project as a whole wouldn't happen if it were just us in the lab. So public support is absolutely essential. With that, thank you very much for your attention, and this is the introduction and background for pollination of American chestnuts with the American Chestnut Research and Restoration Program.
Welcome. So today we're going to be talking about how we collect our transgenic pollen for pollinations and how we freeze or store it until it's ready to be used. So first we have to go on up to our highlight growth chambers here on the ESF campus to collect our pollen. So you can see our trees, they're being blasted with light and given lots of light each day to essentially stress them into producing pollen for us. So that's why they look a little bit sad, but they're serving a very important purpose for providing pollen to us for our pollinations. So you can see me inspecting which catkins to collect pollen from. You're going to want to make sure you collect pollen from the nicest looking catkins, make sure they're not too old and brown looking. So this one looks like a good, healthy contender to collect pollen from. So the next thing that we do is we use microscope slides to collect our pollen. The microscope slides are a really nice option because they allow us to collect really fine amounts of transgenic pollen because we don't have that much transgenic pollen available to us yet. So the slides allow us to go in multiple times, sometimes many days in a row, and keep collecting pollen as the catkins mature. So you can see me rubbing the length of the male flower to collect the pollen. You can see a nice fine dust of golden pollen being collected onto the microscope slide. So the next thing that you're going to want to do is store the pollen in the freezer. We produce our pollen all year round, so sometimes our pollen has to be stored for a few weeks to a few months before we can use it. So in order to effectively store the pollen in the freezer, we need to desiccate it. So when you get back from collecting your pollen on one of these microscope slides, we put them into these slide jars. They're really nice because you can have this nice threaded screw top vial for collecting in the freezer, for storing in the freezer. So you can put up to 4 microscope slides in one of these jars. To desiccate it, you want to leave the lid on slightly ajar for this step, just a little bit open. So you can take one of these canisters. We use a Folgers coffee canister, but you could use something like a Tupperware container as your desiccation chamber. To desiccate it, we use these dry-rite desiccant rocks. They are calcium sulfate rocks. They slowly remove the water from the pollen so that it can be it is safe to go into the freezer. If you were to put the fresh pollen right into the freezer, that could potentially harm it. So using these racks will allow us to safely remove any excess water. So you put a layer of dry right rocks into the canister like this, nice layer at the bottom, and then put your slightly ajar slide jar right into the canister like that, and then put the lid on top. So the next step is to desiccate the pollen for 24 hours in the refrigerator. It is really important that you set a timer. This is a good tip to remind yourself that your pollen is in the refrigerator so you don't over-desiccate it. So you keep the pollen in there for 24 hours, and then after the 24-hour period, you remove your slide jar. And it's really important that you tightly screw the lid closed, and this will create a nice airtight seal for moving it into the freezer. So once you have your labeled slide jars ready for the freezer, you're going to want to store in a frost-free freezer that gets to at least negative 20 degrees Celsius. It is important to use a frost-free freezer because the conventional freezers will change the temperature so that no frost forms, but this could potentially damage the pollen. So after you freezer store it, the pollen will be in the freezer until you're ready to use. So it can wait in there for as long as you need it to be until the female flowers are ready for pollinating. So the next step, once we have approval for the government, is to begin shipping this pollen to you guys at home so that you can conduct your own pollinations with female flowers. So once we have this approval, we'll take our slide jars frozen from the freezer and transfer them into one of these styrofoam coolers that is filled with dry ice. The pollen will be in a bag, probably with a set of instructions for you, but we'll put the slide jar into the freezer, into the cooler, and then it will be mailed frozen overnight to you. When you receive your cooler in the mail, you can take your pollen out and put it into your negative 20 degrees frost-free freezer, and then it will remain frozen there until you're ready to use it. So when it's time to pollinate, you can take your pollen out of the freezer and keep it in the refrigerator while it's in use. So we try to use our pollen that has been removed from the freezer within the first 3 days. That's when it's most viable. So you're going to want to store your pollen in the refrigerator between uses until you are done conducting all the pollinations and pollinating as many female flowers as you can with your transgenic pollen. And that's how you store your pollen in the freezer. Thank you.
My name is Kirla Piccolo. I'm an undergraduate here at ESF studying biotechnology. I'm from Skinny Atlas, New York, and I'm currently working on the American Chestnut Research and Restoration Project. Today we're going to be doing the pre-bagging and pollination steps of our whole pollination process. Now normally we wouldn't do these two steps on the same day. Normally we pre-bag before the flowers have started to fully mature, and then about a week later, once the styles are emerging, we come back and pollinate. This tree is an exception because half of the tree already has styles emerging and the other half has not matured yet. So today we're going to show you both steps on the same day on the same tree. So you want to identify your female flowers, which look like little pineapples, and differentiate them from the long catkins, the male flowers. Here you can see me removing all of the male catkins from the branch where I'm going to be placing the bag. We want to make sure that there's no way that any pollen could potentially clog the female flowers. You can also take off any additional leaves or branches that might be in the way of where you're going to close the bag around the branch. You want to double check that there's no catkins left, and then you're going to grab your pollinating bag. Each bag is labeled with an individual unique number that helps us track which flowers are on which tree and what type of pollen they're being pollinated with. So you're going to scoop up all the leaves with one hand, and with the other hand you're going to put the bag gently on top of the branch. You can see there's a little window so you can check the status of the female flowers without completely removing the pollinating bag every time. You're going to make sure all of your leaves are inside. Then we're going to take a resealable zip tie and use it to close our bag around the branch. Out in the field, we use a cooler to keep the pollen cold while we're pollinating in real time. We want to make sure that we don't take the slides out unless we're absolutely ready to pollinate.
Okay, and this is the pollen you're going to be using up there,
Mhm.
up in the tree
Thank
to
you.
do the pollinating.
Thank you. So now we're going to move on to the actual pollination step. So through your little window in the bag, you can look in and see how far along the flowers are. And once you see styles poking out, that means they're ready to be pollinated. So you can take the bag off. And that's why we use resealable zip ties. Take our bag off. We take the bags off one at a time so we can keep track of the specific bag number, the specific branch, the specific flowers that we're pollinating. So you can see here that these flowers are beginning to mature. And you have the styles from the center nut poking out. And then on either side you can see the other styles just beginning to emerge, but we'll probably have to come back later and pollinate those. This is the first pollination we're doing this year. So those styles on the side are a little bit on the early side. So you take your vial of pollen, labeled with the parent tree it came from. And these hold slides. You're going to take your slide out. And the side of the slide with the pollen on it, you're going to wipe onto the styles. So we're just gently wiping the pollen onto the styles. Make sure every flower gets hit. So once all the flowers have pollen on them, you're going to put the slide back in your vial, cap it up, and then you're going to label your pollination bag with the date, the type of pollen you used, which is on your vial, and the pollinator's initials. In the same way that we did in the pre-bagging step, I'm going to scoop up all your leaves and put that same bag back on the same branch and reseal it. And that is how we do the pollination step. Now I'm going to be showing you how to do the screen bagging part of our pollination process. To do this, we use a wire mesh bag that we fold over and tape the edges so we don't cut ourselves. And then we use a stapler to close the seam of the bag around the branch. We use this to protect from predators such as squirrels that might want to eat our nuts that we've worked so hard to pollinate. To do this, we take our screen bag, we put it over the entire pollination bag, and we secure it with a few staples at the end of the branch. We completely seal off the branch so that no animals can get inside. And that's all we need to do for the screen bagging part of the pollination process.
Well, thank you for participating in this workshop. I hope that you learned many useful skills and techniques and that you can actually help us now with the restoration of the American chestnut tree. If you'd like to learn more about the American chestnut tree and our particular project, please visit our website or contact us at SUNY ESF.